This is a presentation about compliant DNA origami mechanisms being developed at the Design Innovation and Simulation Lab at The Ohio State University. Please contact Dr. Zhou for more details. Scaffolded DNA origami is a recently emerging technology that allows the construction of complex nanostructures via molecular self-assembly driven by Watson and Crick base pairing, that is ATCG. In the past decade, this approach has been successfully used to construct complex 2D or 3D static structures. On the left is the basic principle of scaffolded DNA origami. On the right is a simple example of DNA origami trying to design a simple plate. We expand scaffolded DNA origami nanotechnology to design dynamic nanomechanisms by following a design framework that parallels macroscopic mechanism design. Firstly, we design and fabricate the basic building block links and kinematics joints, for example, revolute, prismatic, cylindrical, universal, and spherical joint. Secondly, we assemble multiple links and joints into structures with more complex motion, which we term DNA origami mechanisms, or DOM. A framework for designing DNA origami mechanisms and machines starts with designing basic joints with motion in specified degrees of freedom. Schematics and TEM images depict joints with a single dominant rotational, or B, linear degree of freedom. In C, DNA origami joints can also exhibit multiple independent degrees of freedom, as in the universal joint, or D, these joints can be integrated into a mechanism which couples rotational to linear motion, as in the slider crank. These mechanisms can be assembled into larger structures that resemble macroscopic machines, as demonstrated by the scissor mechanism in E. The DNA origami scissor mechanism was constructed by first assembling the structural unit on the left and then polymerizing a 1D array, shown on the right, that couples rotational motion of the unit into linear motion of the mechanism. The scale bar in A is 20 nanometer and all the other scale bars are 50 nanometer. The structures in A, B, and D are previous work from our PNAS paper. Recently, we have been focusing on expanding the design of DOM by introducing the macroscopic compliant mechanism concepts into the DNA origami design, where motion occurs by mechanical deformation of components as opposed to traditional mechanisms where motion is facilitated by flexible hinges. We refer these compliant DNA origami mechanisms as CDOM. The compliant components can be designed by placing fewer dsDNA bundles in the component cross-section, which can be easily realized with DNA origami. We have currently successfully designed and fabricated a compliant hinge joint and compliant bistable mechanism. This is about the reversible actuation of bistable nanomechanism. The actuation process utilizes additional ssDNA inputs to fix the flexible structure in the S1 configuration. The closing strands can then be removed via toe-hold mediated DNA strands displacement by adding additional ssDNA inputs, releasing the mechanism back into the free state. Conformational distributions were measured from TEM image analysis, and the free energy landscapes were calculated based on Boltzmann distribution function for the initial free state, the actuated state, and the release state. Representative TEM images illustrate the conformations of the actuated and release samples. The scale bars equal 100 nanometer. Using pseudo-rigid body models for CDOM, we first analyzed the compliant joint with the PRB model as shown in the upper right. A torsion spring with stiffness K was placed at the middle of the compliant hinge segment, and the entire joint became two rigid segments connected by the torsion spring. Furthermore, we also used the PRB model on the analysis of the compliant DNA origami 4-bar bistable mechanism as shown in the bottom. The compliant link was modeled by two rigid links connected by the torsion spring placed near the fixed joint. Then, the energy stored in the compliant link during the motion is equal to the energy stored in the two torsional springs of the PRB model.